Hello viewers, today we are going to see about that introductions about pharmacology. What is mean by pharmacology? Pharmacology they have divided into four categories. The first one pharmacokinetics, pharmacodynamics, pharmacotherapeutics and pharmacognosy. In pharmacokinetics means what the body does to drug. In pharmacodynamics, what the drug does to body. In pharmacotherapeutic means the study of the uses of drugs. In pharmacognosy, identify and crowd materials of drugs, and also pharmacotoxicology will be there. Totally they have divided into five categories, namely pharmacokinetics, pharmacodynamics, pharmacotherapeutics, pharmacotoxicology and pharmacognosy. In history of pharmacology, the father of medicine, Hippocrates, followed by so many scientists, they can design, and first we can use the plant materials, followed by solid samples, liquid samples, etc. What is meant by drug? A drug is a chemical substance used for the prevention, diagnosis, treatment, or a relief of diseases to modify physiological function in human or animals. In pharmacology, the pharmaco means drug and logo means study. Next, the sources of drugs. They have divided into so many sources will be there, namely plant, microorganism, animals, minerals, recombinant DNA technology, etc. So here majorly say that six major sources of drugs will be there, such as plant sources, animal sources, minerals or earth sources, microbiological sources, semi-synthetic sources or synthetic sources, and recombinant DNA technology or rDNA technology. These are the main six major sources of pharmacology. Now we are going to discuss about that one by one. Generally, the source of drugs divided into two types. One is natural and second one synthetic. Natural sources namely plant, animals, minerals, microorganisms and human. In synthetic sources, chemical synthesis in and other techniques namely cell culture, recombinant DNA technology and hybridoma technology. The first one, the main common sources of drug namely plant materials nowadays used for predominant sources of natural drugs. For example, leaves, bark, fruits, seed, etc. The plants having two types of metabolites. The first one primary metabolites, namely carbohydrate, protein, vitamins, minerals, etc. And secondary metabolites named as alkaloid, flavonoid, tannins, saponins, steroids, glycosides, etc. In secondary metabolites, the plant produce the alkaloids, glycosides, tannin, hotel oil and resin, etc. These bioactive compounds mainly used for therapeutic properties. In second, animal sources, the animal sources, so many variety of drugs are derived from animal tissues or secretion. For example, hormones, example insulin, enzymes, pancreatin or pepsin enzymes, biological, it is a vaccination and anti-serum, And last one, glandular products, especially examples for thyroid extract. These animal sources especially used for therapeutic uses, that substance are critical in treating certain disease, for example insulin for diabetes. And the third one, marine sources. The marine sources rich amount of bioactive compounds in unique and novel biodiversities of the novel bioactive compounds will be present, especially for the marine sources. For example, sponge, corals, algae, marine bacteria, and fungi. These algae or fungi of marine sources especially can produce anti-tumor, anti-inflammatory and antimicrobial agents, especially used for that. For example, cytotrabin drugs especially used for anti-cancer properties. It is one of the marine sponge. So the big challenges will be there. The difficulty from collections and cultivations of marine sources. It is a very very challenging, and also the historical slowed the discovery and mass productions of marine derived drugs, but advance in aquaculture and synthetic approach are overcoming these sources. Next, tissue culture. Generally, the tissue culture divided into two types: plant tissue culture and animal tissue culture. In plant tissue culture, we can use in vitro techniques that involve growing plant cells, tissues, or organ in nutrient media under sterile condition. The very important allow for the productions of secondary metabolites, that is bioactive compounds, without a needing to cultivate whole plants in the field, and enables micropropagations of rare or endangered medicinal plants. And last one, potential for genetic transformation and metabolic energy, metabolic engineering to enhance yield of therapeutic compounds. Next is the synthetic sources, especially the synthetic sources used for the chemically in laboratories. For example, paracetamol, aspirin, and sulfonamides, etc. In synthetic drugs or semi-synthetic sources, in synthetic sources, when the nucleus of the drug from natural sources as well as it is chemical substance or structure is altered, is synthetic. For example, bismuth iodine. The semi-synthetic sources, when the nucleus of drug obtained from natural sources is retained but the chemical structure altered, we call it is semi-synthetic. For example, ethyl, ethylene, testosterone, methyl testosterone, ampicillin, etc. In microbial sources, especially for Penicillium notatum is a fungus which give penicillin. As Actinobacteria give septomycin. Aminoglycosides such as gentamicin and topromycins are obtained from septomycins and micromonospores. And recombinant DNA technology or rDNA technology involves the cleavage of DNA by enzyme restriction endonuclease. The desired genes is coupled to rapidly replicating DNA. For example, viral, bacterial, or plasmids. The new genetic combinations is inserted into the bacterial culture which allow productions of vast amount of genetic materials. Examples of drugs, for example, acetyl salicylic acid, it is a chemical name. Aspirin, it is a genetic name and a non-proprietary name. In brand name, disprine. And the second one, acetomorphine, it is a chemical name. Genetic name paracetamol and brand name Colpal, Metazine, Crocin. And the third one, benzene, it is a chemical name. Ampicillin, it is a generic name. And rosin, it is a brand name. These are the some examples given for the chemical name, generic name, and brand name. All the chemicals having the three important names. So this picture shows the route of administration for especially injection position. Intramuscular 90 degrees Celsius, subcutaneous 45 degrees Celsius, intravenous 25 degree, and intraterminal 10 to 15 degrees Celsius. So degree we can inject it into the liquid injection solutions. Angle, especially we say that angle of injections. So generally, what we are covered this PBT, especially for what is meant by pharmacology. So the pharmacology means understanding the therapeutic potential, analyzing the safety profiles. It is a broad science studying how to substance, that is drug, interact with the living organism, encompassing their properties, action, and effort. What is meant by drugs? Any chemical substance that, when administered, produce a physical or psychological changes in living organism, excluding nutrients. And dose and response, we have a three type of dosage: therapeutic dose, toxic dose, and lethal dose. ED50, ID50, LD50. The main branches of pharmacology name is pharmacokinetics, pharmacotherapeutics, pharmacotoxic, pharmaconazi. These are the main important branches of pharmacology. And what are the side effect and adverse effect? Side effect, we have desirable that, for examples, if you are continuously prolong take tablet means we have affected for the inside organs. In adverse effect, it is a forming of allergy formations. Next, drug action, we have receptors, go to the agonist to antagonist. So this way of drug actions will be there. And the route of administration, so many route of administrations will be there, and mainly oral, injection, topical, and inhalation. So what are the role of drug? So mainly ADME properties in pharmacology work. So drug will absorption, then enter into the blood, then enter into the target organs, and we can effort metabolism and excretion. That means ADME properties. A for absorption, they can dissolve into the water or acid, then we can distribute it to the whole body, that is D for distribution. Third, for metabolism, that is divisions of that metabolism reaction we can cultivate it. For example, carbohydrate is converted into glucose 6 phosphate, glucose 6 phosphate, fructose 6 phosphate. This the metabolism reactions will be occur. And finally, what the pains, whatever we have present the pain, so directly go on to the rectify the pains, then we can eliminate it or excretions. These are the main important points. So the pharmacology mainly work on it, the four important points. First one is absorption, second one is distribution, third one metabolism, and fourth one excretion. So these the main important functions. So thank you. I hope you can understood the basic introductions about pharmacology. Thank you.